All right, hi. So this video is going to go through A2. And A2 is interesting because it does actually have 3 different components, but 2 of them are HL only. So I'm going to go through them in order. So if you're SL, you might want to skip ahead a little bit and then just watch the middle section. So this first component of origins of cells is HL only. So we're going to talk through that first, and I want to make this kind of essentially flow chart that looks at kind of the steps involved to go from a prebiotic earth all the way down to cells. So I'm going to you can kind of set up your page. So I actually want to go ahead and show it all to you first. So this can take up your whole page. So this one's going to be a little bit lighter. Lots more, but this one's not so bad, so we should be able to kind of use up our space and space yourself out. But I'm going to go ahead and go down the left hand side and kind of go through all of this. So what I want to look at is kind of this whole idea of the origin of cells. We started with what's called prebiotic conditions. So biotic meaning living. So we need to look a little bit at what were the conditions that could have given rise to the start of this process. And then from that kind of prebiotic or abiotic environment, the first thing that had to evolve was monomers. So we needed to then get biological molecules of some sort, so not all the way to living things. So we'll go prebiotic conditions, and from prebiotic conditions arose monomers. Then from those monomers, they had to assemble into polymers. So monomers became polymers. And then those polymers had to organize into cells. And then from the kind of earliest cells, we'll then end on the last universal common ancestor, or LUCA, which is like from the first cell. It's not the initial cell, but the last of the initial cells that you can still find evidence of in living organisms now. So I'm going to erase the board and finish with kind of cells in LUCA. So cells, and then we will have kind of one more line down here for LUCA. So this first part is kind of the first half of your page, and then I'll erase and keep going. But the goal was to kind of try to organize that understanding. So when we look at kind of where does all this fit in, to try to put it in some sort of kind of concept. So when we say prebiotic conditions, let's go through what those conditions were that are significant. So the first thing we want to know is that there was so the conditions included a lack of oxygen. And related to that is also, therefore, a lack of ozone. So ozone is O3, so it is an oxygen-based molecule. And that's going to be important because that means that UV is going to be able to penetrate, and therefore the temperatures are going to be much, much higher than we have now. So once oxygen formed, it created the ozone layer, which reduced kind of global temperatures. Next thing was the gases were different. So we did not have the oxygen, but in place there was high levels of carbon dioxide and methane. And then related to kind of what we talked about with ozone, there was high UV penetration and therefore high temperatures. So what we're trying to look at is it possible for those conditions to then give rise to life? And so evidence says yes. So evidence says abiotic conditions like these could spontaneously produce living monomers. And that keyword kind of spontaneously, so it is something that could happen on its own if the conditions were right. So produce biomolecules. And of course, we're going to look primarily at first producing monomers. So I'm going to draw a tiny Miller-Urey experiment. You can see I just drew a little one to represent kind of the evidence for that. So this is kind of the statement, And then the question is kind of what is the evidence that that is true? And the evidence for that is going to be the Miller-Urey experiment. So we drew in class in our booklets a bigger version of it. So this is going to be a small version of it, but I think it's probably all you would really need. So there was basically two kind of larger chambers that are going to represent important parts of the, this is a bad drawing. And then they're going to meet here in this like kind of collection chamber place. Okay, so there's my very rough drawing of it. Over here, this is going to represent the oceans. So here we're going to have water and it's going to be boiling. So they had to put kind of heat under it. So that's going to be my boiling water. And again, that makes sense because we knew there was high temperatures. So that suggests the oceans could have been in a state naturally of boiling. And so as it boiled, that was releasing vapor. So we're going to have water vapor being released. And then over here, we're going to have our gases, and that's going to represent the atmosphere at the time. So the gases that they have are going to try to represent some of those things. So they had methane, they also had quite a bit of ammonia, and they're going to have some hydrogen. So gases that were similar to what they believe would be found at the time. And then the third kind of component we didn't put in our list, but it was believed that there was more natural electrical activity or lightning. So to represent that, they also added electrodes to it. So there's my boiling water, my gases, and my electrodes. So it represents oceans at warm temperature, the gases we believe they were there, and the idea of the kind of prebiotic atmosphere. Then there's a condenser, because all of this is gaseous. So it's going to be the water vapor mixing with the other gases. And so then that should now form a new kind of liquid, which is going to be primarily water, but mixed in with the water are going to be these molecules. And so they tested this solution, and that is said to be the primordial soup. So primordial soup is basically the water mixed with biomolecules. And the main biomolecules they found were amino acids, and what looks like they were probably the start of fatty acids. So interestingly, they did not actually necessarily see like RNA nucleotides, but it's possible that that was because they didn't have a phosphorus source. So if we did kind of repeat that and change conditions and look at adding some sort of phosphorus source, we could have seen RNA nucleotides as well. But it did show that you can go from abiotic things to the formation of biotic elements, or biotic compounds. So that was kind of step one. So step two is that we need to then look at the idea of, can those monomers self-assemble into polymers? So they would need to self-assemble. And so if you go back and think about what we talked about in B1, we would say, okay, well, self-assembly is a condensation reaction or dehydration synthesis. So yes, it can happen by removing water, but a lot of the times it revolves around some sort of catalyst. So in our body, we have enzymes that act and do that kind of job. So then the question becomes, how do you manage that without enzymes? So the first kind of challenge is that you need a catalyst for that kind of self-assembly to happen. And so the theory is, if we need a catalyst, normally proteins act as catalysts, but then we're kind of looking at a circular argument, because how do they self-assemble into an enzyme which is structurally complex without a catalyst to get to the enzyme in the first place? So that's kind of the debate. So the theory here is that what could be the catalyst could have been RNA. So instead of kind of starting with proteins and DNA, the argument would be that we could actually have started with RNA. So the RNA could predate DNA. And you'll see different titles for that. So your syllabus doesn't use the term RNA world, but sometimes you'll see that as just a name for that theory, that RNA was actually the first of the biomolecules for nucleic acids. And so the question then becomes, well, what is the evidence for this? So if we're going to say that's the case, so just like we said above, we have the evidence is the Miller-Urey experiment. What is the evidence here? And in this case, the evidence is some of the features of RNA. So there's kind of three reasons why this is thought it could be the case. So first of all, if we were to kind of assemble a biomolecule, RNA is really small. So we're looking at usually just assembling a few nucleotides together compared to either a DNA, which has the whole double helix structure, or a globular enzyme. So it's structurally, so it's single-stranded and small. And so that means it's more likely to be able to self-assemble. So that's kind of the first argument, and this is kind of why do we think it might have predated DNA. So if we compare that to DNA or to proteins, either of those are much more complex. Compared to DNA, so if we were looking at DNA, RNA does have the ability to act as a catalyst. And the reason why we know this is because there is RNA inside of ribosomes, and that RNA inside of ribosomes plays a catalytic role in translation. So we call those ribosomes, like instead of enzymes, they're ribozymes. So ribozymes, and so they actually catalyze protein synthesis. So we know that this is possible for RNA to do this. So we can see evidence of this happening now. So that tells us that RNA is, so all of these are evidence for RNA. So RNA is simple, RNA can act as a catalyst. And the other thing that RNA can do is it can actually provide the template for two different things. So RNA can act as mRNA and make proteins, and it could also be copied to make DNA. So one thing could actually make two other biomolecules. So it can actually be used to make proteins and DNA. So for those reasons, the theory is that RNA played this key role, and if we were able in our primordial soup to have some RNA nucleotides, they would be able to self-assemble and then act as catalysts for the self-assembly of other biomolecules as well, which is really important. Okay, so now we're starting to have polymers. So now we're going from our primordial soup with individual monomers to these decent polymers. But the next question becomes, okay, well, how do they become contained in like a cellular unit instead of just a soup of watery compounds? And so essentially what needs to happen is we need to form a membrane. So we need to start to actually isolate them inside of membranes. So our next step to go from a polymer to a cell is the need for formation of membranes. If we start to be able to take water mixed with polymers and surround it by a membrane, that basically creates a cell. So a cell is a cytoplasmic solution with a membrane around it. And so the idea is, what is the evidence that that could happen? So the argument is that this is believed to be possible. And the reason it is possible is because of the tendency for amphipathic molecules to naturally orient in a circular. So the tendency for the formation of rings with amphipathic molecules. So the argument is here if we go back to the primordial soup, we know that in our primordial soup, we believe there were fatty acids, and there would have been amino acids and other kind of molecules. So again, probably what we're missing is some sort of source of phosphorus to get our phospholipids from that experiment. But presumably if there was kind of a phosphorus source, we could form either double layers like our traditional phospholipids or even a fatty acid on some sort of polar tail. And if that happens, they will naturally kind of try to face like this. So this would be a natural tendency for them to form kind of a circle. So that argues that if you have in solution amphipathic molecules, this will happen on its own. And so again, our question is, okay, well, where's the evidence for that? How do we know that would happen on its own? And the evidence is we can basically experimentally see this happen. So if you put phospholipids in solution, they will do that. And we call that spontaneous vesicle formation. So this is saying this automatically happens to form vesicles. And that same process could be occurring on a cell membrane level as well. So we can see it happen on a smaller scale, and arguably the same thing could happen on a larger scale. And then basically kind of between this and there, so that would kind of give rise to what we would call protocells, which are, you can use the word two different ways. One would be like a kind of artificially made fake cell, and then the other would be essentially earliest cells, so not true cells, but these would kind of be the precursor to cells. And then to go from that to actual cells, you would need to, the last kind of step of that would be to actually have some degree of compartmentalization. So to actually go from just kind of having a whole bunch of macromolecules inside a solution to start to kind of organize it into some sort of function. Okay, so I'm a little bit more than halfway through my page, but I'm going to erase to do the last part. But we're going to keep going with this same concept. Okay, so if we keep going, we're now at cells, like so. And then we are going to, the last thing is from cells. So we kind of said origin of cells, but then from the first cell, there would have been kind of a bit of further evolution to get to the last universal common ancestor. So this is an organism or a cell that shares features with what we still have as living cells today. So kind of from the very first formation of a cell all the way through. Okay, so while we're on cells, let's just use that as a little opportunity to quickly establish what does it mean to now be a cell. So how have we gone from essentially kind of protocell through to a cell itself? And so a cell is the smallest unit of life. To carry out all functions of life. So this is one thing that can do all the different things that are needed to be considered living. So let's look at what are the functions of life. If you look at these different places, some will give you 6, some will give you 7, some will give you 8. So we're just going to write the 8 that the IB syllabus puts there because, you know, if that's what they're telling us, we'll roll with it. So the 8 things that they say are the functions of life are that it maintains some degree of homeostasis. So the ability to maintain a constant internal environment. The most obvious one that all cells do would be water balance via osmosis, which more on that to come. They have some sort of metabolism. So one way you can think of that is that they do have to essentially create their own ATP. So that's kind of the most basic thing, is that all living organisms do some sort of cellular respiration, which is a metabolic process. So if they do cellular respiration, make their own ATP, that is covers that. Excretion, so some sort of removal of waste, which sounds fancier than it is. It does not mean they actually have to like release urine. They just can exit things through the cell membrane, but they have some way of dealing with waste products. They have some movement. That one's a little bit tricky. If you think about plants, the whole organism does not move, but things move within the cell. So think about like endoplasmic reticulum. So there is transport of materials inside the cell. So it doesn't mean the whole organism, but there is movement of materials, is a good way of thinking of that. They have to have some sort of nutrition. So photosynthesize or take in food. They have some ability to grow, somewhat limited with unicellular organisms, but they do usually divide in two and then have a growth period. They have some ability to respond to stimuli. So they are kind of aware of their environment and respond to situations in it. And they have the ability to reproduce independently. So those are kind of my 8 conditions that are carried out by a cell. Let's look at kind of the idea of eukaryotes definitely do this. So easy peasy lemon squeezy. Prokaryotes, bacteria in other words, yep, they can do all of those things even though they're unicellular. Who cannot do all of those things? Viruses. So viruses are not able to do all of these things. So just some examples. They don't have anything to maintain homeostasis with. So basically almost all of them. They have some DNA, but you can see they don't meet those kind of requirements of life. While we're on the requirements of life, the ability to carry all of that out is aided by the process of compartmentalization. So this is just one of the vocab words in this section, which is the ability to have some organelles do different things. So instead of having every organelle do everything, the idea of assigning it. So if we, even in prokaryotes, they start to have like the cell membranes in charge of homeostasis and excretion, but then the cytoplasm might be in charge of growth. So you start to kind of assign regions, functions by region. It's a very short explanation, but the whole cell doesn't have to do everything. It can start to kind of divide it up. Okay, so then between cells and last universal common ancestor, there's been evolution and extinction. So we are not talking about cells that there is no kind of current existence of. So we're looking for what are some potential ancestors that are held, kind of common to everything that's still living. So let's have a look at, we're going to make our little branch. I think we made one similar in class, but honestly, it's just kind of the best way to explain what this is. So this might be modern prokaryotes or bacteria particularly, is one branch. And then from there, there's been kind of a branching out of a different pathway, and it divided out twice and created these kind of extreme organisms called archaea. More on that in like A3, something like that. And then eukaryotes. So all living organisms, and then all the way down here might have been, goodness, sorry about that, the first ever cells. So here's my first cells, and then a bunch of things came extinct. And so where we're placing Luca is right here. So the last kind of living organism before things branched off. So the last time it was common to everything, that's our universal common ancestor. So again, some of these we're not going to count because they became extinct, but the last one that was there and before the kind of divide off went. That's Luca. Let's look at what is the evidence for, and also a couple things. How old is Luca? If you Google how old is Luca, it will tell you that Luca is 13 years old because that's a Pixar movie. So, but actual Luca is estimated to be about 4.2 billion years old. Okay, years old, that's why I'm abbreviating. So let's look at kind of some evidence and then we'll end with how did we get to that amount. So what is the evidence that Luca does in fact exist? So what is the evidence that you could in fact find an organism that is a common ancestor to all living things? So we're gonna basically look at kind of 4 pieces of evidence that will cover the main points. The first thing is just the general evidence of the fact that there is a universal genetic code. So if all of these different organisms evolve separately, so bacteria evolve separately to eukaryotes, what is the odd that they would all end up whereby CCC is proline? Like, seems very unlikely. Similarly, there are 300 genes that are found in all living organisms. So we have a whole lot more genes than that, so it's not that many, but there are 300 kind of conserved genes that they found across archaea and eubacteria and eukaryotes. And kind of related to the similar genetic code, we have the same basic biomolecules. So you will actually see kind of bacteria use glucose, create phospholipids, so all of those same kind of biomolecules, and similarly kind of same metabolic processes. They're not all the same, but when we get to anaerobic cell respiration and we talk glycolysis, it is a process that happens in every living organism. Okay, so if we have these 300 shared genes, one of the things they did was try to look at what are they for, and genes are involved in anaerobic processes. So those are processes that take place in low oxygen environments. So if you go back to kind of prebiotic Earth, that makes sense. If Earth started with prebiotic conditions, it would be logical to have genes that helped kind of establish in those conditions. And then if you say, okay, well, where is there still low O2? There's still low oxygen in hydrothermal vents. So one idea is that the last universal common ancestor came about from an environment like that, and we might in fact still see kind of very closely related organisms to Luca if we look in hydrothermal vents. So it might be that Luca if we wanted to figure out where would we find it, we would look in hydrothermal vents, and when we have looked there, we have found lots of really old fossils, which suggest, yes, that might be the case, that that earliest life might have arose there. Last little thing is related to the aging. So if we said this is how old we believe Luca is, so the question is, how do we age fossils to tell that? So we said there's old fossils, but then how do we age them? I mean, just pretty briefly, because all the syllabus says is like, understand that it's old and that there's different ways of dating it. It does not say you need to go into radioactive labeling and so forth. So I'm going to do super short version of it because I don't see it likely that they could ask any complex question on it. So there's 2 ways to date fossils. The first one, it would be absolute dating, and then I'm going to put that on the bottom. So I'll put relative first. So relative dating, and then the second one would be absolute dating. So if we say relative dating, we are just looking at approximately compared to other fossils. So we would compare kind of its placement. So where is it placed in rocks? And we would be comparing that to other index fossils, which are going to be fossils that we actually know some information about. So we would say, okay, I know that this fossil is this many years old, and so if it's on the same layer as that. And then absolute dating is going to be, we can actually work out its actual like age in years. So in order to get a number like that, we're going to use the half-life of radioisotopes. So we can measure how much is still there and how much would have been there in a fossil of that size initially, and use that to get a sense by knowing how long it takes to have it, of how long it would take. So I'm not going to go into a lot more detail because again, I don't think that's a heavy component, but that's just a short kind of thing about how we could get to that age. Okay, so next part is going to be for SL as well. I'm going to erase the board, and then we'll be doing cell structure. It's, yeah, a lot, so get a blank sheet. Okay, so I set it as a maybe arbitrary goal that I wanted to do each subsection, like A 2.1 versus A 2.2, on exactly one sheet of paper, just as my way of trying to make sure I'm summarizing it. Having said that, by the end, I had to write really, really tiny. So you may want to just make your own piece with writing it on one side, front and back. If you also are trying to write it in one side, you might want to write smaller up here, because I was all like writing big and then had to write lysosomes in like 0.6 font. So we're gonna, it does fit in a page, but it will be tight. So A 2.2 is the only one that's standard level, but there's quite a bit there. So there's all the microscope part, and then there's all the cell types, and then a little bit on every organelle. So, and then there's also a little SL part that we'll add on, looking at endosymbiosis. So, I mean, HL part only. So let's get started. So if you look at the syllabus, there's a few things that are hard to conceptualize how, sorry, you should take that off, how they would actually ask you about it. So there's a lot on like microscope skills, but obviously they can't really ask you in an exam to like use a course focus. So hopefully we've covered that in class, but they could ask you about different types of microscopes, or they could ask you, and they will ask you, about magnification. So let's kind of talk a little bit what you could have to know for an exam on microscopes. I'm not going to draw a microscope and label the parts. That doesn't seem to necessarily be in there, other than the skill of using one, and hopefully you've done enough with that you know how to do those. So if we're going to say cells, where does that fit in? So cells, how do we actually visualize cells? We're going to visualize them with microscopes. So we are not just kind of making this stuff up in terms of like, this is how it looks. We outdo between light and electron microscopes. We do have actually really good understanding now of what components of cells look like. So how do we visualize them with microscopes? Okay, so you need to know about the two types of microscopes and a few things about kind of technology or advancements within those two types, and then the calculations. So we'll start with kind of the two types of microscopes first. So we have light microscopes, which are what we use in the classroom. And then there are electron microscopes. So short version of the difference between them. With light microscopes, I think it sounds pretty straightforward. The light is providing the actual source. The problem is with the light, the way it works is as you try to go up magnification, the long wavelengths cannot preserve a resolution. So they have lower resolution at high magnifications. So as a result, we are pretty limited in how high of magnification they can go. So they usually max out around 1,000 to 2,000 times. Some really good ones can hit 2,000. General, relatively affordable ones usually can't go of about 1,000. But some advantages are you can use them with living organisms. So you can actually view organisms in motion. And the other thing is they do provide color, which is nice because it can, if we use stains, kind of help us actually visualize some things. And we'll come back to the color in a minute when we get to some of the advancements. Electron microscopes, on the other hand, they pass electrons through it. Electrons have a shorter kind of distance and actually preserve resolution. If you are a physics person and I just gave a really bad explanation for that, it's fine. You don't need more than that. At a bio level, you just need to know light, low res. Electrons, high res. So these preserve resolution. So by doing so, we are therefore able to have higher magnifications. And that's really the main benefit of an electron microscope, is that we can get these much higher magnification images, which is going to allow us to actually see inside of living organisms. Inside of dead organisms. So no living organisms. And they also do not generate any color. They're also crazy expensive. So I'm just going to draw a bunch of dollar signs. So they're not really doable in a lot of laboratories, so like school laboratories, but they are used in kind of big scientific labs. So they're able to be accessed for, with a lot of funding, but you won't ever be able to look at one in a school setting. Okay, and within the electron microscope, there's two main categories: scanning and transmission, which you'll often see as SEM, TEM. So scanning electron microscopes are only going to look at the surface. So they're going to provide a pretty good 3D image of the surface. A transmission electron microscope can actually go inside. So this can actually allow you to view the interior, but it's not going to give as cool of a kind of 3 dimensions. So they both have benefits. Okay, within both of these, you need to know a little bit about some new advances. I can't imagine this would be more than a multiple choice question here or there. Who knows, maybe they could ask you like a 3-point question about some advances, but you don't need to know any of them in like phenomenal detail, just that they are kind of advancements in this area. So in terms of light microscopes, they have fluorescent light microscopes. And so fluorescent microscopes actually use like high UV light instead of a normal light. So they're going to use kind of high spectrum lights. So they're going to use high UV lights paired with some sort of fluorescent stain. So there's kind of 2 different options. You can just use fluorescent dyes that stick to an actual cellular structure. So that could be a stain that sticks specifically to like the nuclear membrane. So those would be stains that kind of adhere to one structure. And that would allow you to specifically visualize that one structure. And then the newer kind of even more advanced to that is called immunofluorescence, which just means it uses antibodies. So instead of having to actually have the stain stick to it, which could be a little bit trickier, it's essentially dyed antibodies that then bind to a structure. And these tend to work for smaller structures, including things like a protein. So it can actually bind to a very specific protein. So instead of it just sticking to anything, you can make it look for like, are there viral proteins involved? So this is kind of a really cool kind of further advancement in this area to be able to kind of create that. So you can have like a little antibody like this, and this could be specific to one exact protein. And so if it binds to it, attached to that is some fluorescence. And so the actual antibody binds, and then that fluorescence attached to the antibody is what shows up. So that's kind of a cool little area of improvement to let us like isolate more specific particles within the cell. And then the 2 things for electron microscopes to know about is freeze fracturing, which basically sounds like what it is. So it actually freezes and then fractures or cuts it across a plane. So it allows you to etch a specific plane and then view the etching under the microscope, which basically would allow you to kind of essentially cut open an organelle. So if you fracture it across it and then open it, you can kind of see exactly what the interior would look like. And it's been really important for kind of looking at membrane structure because it's allowed us to actually kind of open the membrane at different points and actually visualize its membrane and also the membrane of different organelles. Again, you don't need to necessarily memorize some complex thing like freeze it, fracture it, etch it, view it. So that's probably about all you would need to say about that. And then cryogenic electromagnification, which is generally abbreviated as cryo-EM. And so basically they freeze a protein grid. So frozen protein grid, usually not in water, but in like ethanol. So freeze it in something like that. Then you put it under a electron microscope and you can see the way that the different components of that protein transmit the electrons, and that is based on its structure. Yeah, so that's frozen protein grid, and then you can track the electron transmission patterns, and then use a computer to generate a structure. So the computer will generate based on how the electrons kind of were transmitted when it's observed in the frozen grid under the electron microscope. That will give you an image of that. So that's as much as we're going to go into because I am not a up-and-coming microscope expert, but if you are excited by that, I am sure there are some fantastic TED Talks about it, but this will be enough for what you need to know for the exam, hopefully. Okay, so then they could ask you about the types. The other kind of skill set when we look at this is that you could have to do calculations. And generally the calculations are either to calculate a magnification or to then calculate an actual size. And so I'm going to show it to you as a two-step process. If it happens to give you some part of this and it's easier than this, great. But most of the time it's a two-step process. So it's going to be step one, calculate the magnification from a scale bar. So on the image, there will be some sort of scale bar, and using that, you need to calculate how much it was magnified. So the formula for this is that the magnification is going to be the actual size of the scale bar. So scale bar by ruler. So you need to actually measure it. So you measure that top one. And then divided by the number on the scale bar. Sorry, it always says like actual size, image size, and I always find that confusing as to which one are they actually referring to. So the top number is what you measure, the bottom number is what it tells you. Be really careful because when you're using a ruler, you're most likely getting it in millimeters. So remember that for every 1 millimeter, that is 1,000 micrometers. So you will have to convert your units because most of the time what it actually gives you in the scale bar is in micrometers and you're measuring it in millimeters. Okay, so then that should give you the actual magnification. So then step 2, you are going to use that magnification to actually calculate the actual size of the object. And so to do that, you are going to say, so the actual size is going to be the actual size in pick by ruler. So So anytime I say by ruler, that means you have to measure it with a ruler over the magnification. And then that will give you the actual size. So this is the actual size here, and you have to do that. So most of the time you're gonna have to calculate the magnification first, then you're gonna have to do that. So be careful, try to be really precise when you're using your ruler, try to get as good of values as you can, and try to be precise and not just round too much, or you can get wrong answers, but that's kind of the general idea of it. Okay, so that's all things microscopes. So what I'm gonna do now is we're gonna make a table of organelles or structures. So leave a little bit of room on the side if you're trying to fit it on one page, because I still need to go through a few other things. So I'm gonna have my table be like the majority of the page, but leave a little sidebar for 2 more things. So after we do all the organelles, we need to go through atypical eukaryotic cells and endosymbiosis, and we're gonna stick that over on the side. I think it's the easiest way to go. So just a little bit of formatting information for you. Okay, so what I want to do, so in class we actually drew the organelles. So we already cut the cells and drew each organelle inside of it, and we drew like 3 different cells and showed their organelles. So you already have it done that way. So just to keep it different and maybe a little bit more consolidated, we're gonna go through the cell structure. I'm using that term because IB is really picky and says like cytoplasm's not an organelle. So I don't want to be inaccurate. So the structure of a cell, we're gonna then, I'm gonna use a color key, so I'm gonna put it like everything in red that are in all cells, so you'll follow on. And then we're gonna write a really brief function. So when we get to B2, there'll be more kind of organelle function, but A2 does say that you could have to provide a statement of its function, so it doesn't need to be detailed, but we do need a 1-line function. Then we're gonna clarify what type of cells is it found in, particularly if there's any sort of difference. And then we're gonna do a like very simple diagram, but it does say in this syllabus that you could have to create a diagram or be able to draw from an electron micrograph. So we're going to try to draw as much as possible a little diagram that shows what it might actually look like on an electron micrograph. Remember that I am accepting my limitations, so this is for my class. You do have this key, so you may want to have this out next to you and be like, oh, that's what it's actually going to look like. So worth having a look at this. If you're not in my class, this is in Oxford's textbook, so it's another just a good tool to kind of look at what these things actually would look like under a micrograph, or which is a microscope image. Okay, so moving along. So we are now on cellular structures. And we're gonna do this as a chart. I didn't end up drawing like the vertical lines because I didn't do a perfect job of like keeping it straight. So they ended up just being straight lines across. So whatever works for you. So we're gonna say, what is the structure? Then we're gonna say, what is its function? Then we're gonna say, what kind of cells is it found in? And then we're gonna draw an adorable tiny pig. And then notice I didn't go all the way to the end, so I still have some space here that I'm going to be able to add some other stuff in. So I'm going to start with the cell structures that are found in all cells. This is just also a way of revising kind of what things are common between prokaryotes and eukaryotes. So there's 4 things that are common between prokaryotes and eukaryotes. So I'm actually going to start with saying there's definitely not an organelle, but it is something found in cells, which is DNA. So every living cell has DNA in it. And again, I'm just going to use red for these first 4 to be like, everything has it. What is its function? We should all know this now. It is instructions for proteins. And it is found in all living cells. But when there's a point, I just want to make it clear a little slight difference. So I do want to make a note when there is a distinction between prokaryotes and eukaryotes. So for prokaryotes, it is circular, and it's found in the cytoplasm. In eukaryotes, it is linear and it's attached to histones. So I'm just going linear and wrapped. This table's already giant, so I'm gonna have to do a few little shortcuts as we go. In terms of, therefore, what does it look like? So if we see it for prokaryotes, it will usually be a jumble, but it should meet back up. If we see it for eukaryotes, it might look like a long line. If it's condensed into chromosome form, it may look a little bit more intense. And the reality is, if you're seeing it on an electron micrograph, it's likely to just look like a dot. So a little section in the nucleus. So it may not look like much of any of that if you're looking at it under a microscope, unless you're seeing it in chromosome form. DNA, done. Woo, woo. Do a better job with your lines than me. Well, that's really stressful and sad. Oh. All right, gonna have to move on. Okay, next. Next thing that's in all cells, use a ruler, is going to be cytoplasm. So next up is cytoplasm. What is the purpose of cytoplasm? Is to basically just contain the other organelles. And there are actually a number of chemical reactions that happen in the cytoplasm, particularly in prokaryotes, because they don't have the same level of compartmentalization. They do tend to do more reactions in the cytoplasm. The cytoplasm is found in all cells, and the cytoplasm doesn't really look like anything. It is just the interior of the cell. So it is just everything inside. So it's just the inside of the cell. So it's not going to have any amazing structural appearance. Okay. Of course, what contains it is the cell membrane. So next up is going to be a cell membrane. So there are other things you can say for the cell membrane in terms of being like the outer boundary and so forth. And sorry, I know I'm running a bit small, but I think you can just zoom in on your screen and it work. But the most important function of the cell membrane is going to be to determine what enters and leaves the cell. So it regulates entry or exit from cell. So it allows things to go in and out of the cell. So it is the kind of semi-permeable membrane, and that's really important. And it is also found in all cells. If you're looking on an electron micrograph, you're not going to be able to see very much other than a line. So if you're looking on a microscope, it's just going to look like that. But just a reminder, what does it look like in reality? It is a phospholipid bilayer in all living organisms. So there's our little phospholipid bilayer as a reminder that that's what it actually looks like structurally. Okay, the fourth thing that is found in all cells is going to be ribosomes. So ribosomes are up next. The purpose of ribosomes is to make proteins. So that's going to be protein synthesis. Ribosomes are also, like we said, in all cells. But like DNA, we are going to make a note that they do appear different in prokaryotes and eukaryotes. Their difference is based on their size. So in prokaryotes, they are smaller, so they're called 70S, which is just a measure of their size. And in eukaryotes, they are larger, they are 80S. It's like the two subunits. You just can know it as 70S and 80S is what the syllabus has it written as right now. If you see an electron micrograph, it's likely to just basically appear kind of like as a circle. But reminder that in reality, it is kind of like that. So it has the kind of two subparts that come together. Okay, that is the end of all cells. There is one more that is shared between prokaryotes and some eukaryotes, and that is the cell wall. So it's gonna end up being its own color because there's only one thing in this exact category. But my cell wall, it's not just prokaryotes, it's not just eukaryotes, but it's not all cells. So it's its own little special category. And it's going to be for shape, structure, and support. It is found in 3 types of cells that we want to know. So the 3 types of cells that it is going to be found in is going to be plant cells, fungi, and prokaryotes. So in eubacteria in particular, and I'm going to move this over slightly. And you do need to know that it's made up of a different compound. It's made up of peptidoglycan, pepti for protein, glycan for carbohydrate, which is kind of a carbohydrate protein molecule. In plants, and you should know this one already, it is made up of cellulose. And in fungi, it is made up of chitin. Looks like chitin, actually chitin. Okay, and then if we were to draw the actual picture of it, so if we were to look at, again, here's my cell membrane, like so. It's going to sit on the outside of the cell membrane, so it would look kind of like that outside the cell membrane. The way I've drawn it is thick layer, so that is an example of what it would look like in gram-positive bacteria. It's like a tiny little dot point in the syllabus. The dot point really just says that you need to know what the cell wall looks like in gram-positive, not other weird types of bacteria, which is basically just telling you that you can just learn peptidoglycan is what prokaryotes have. In reality, some other prokaryotes have weird other things, but you can go with bacteria have peptidoglycan. That is true of all gram-positive bacteria, not necessarily all, so just kind of a little bit of awareness of that. All right. From there, I'm going to put these 3 all kind of together because I didn't want to give them that much space on my chart. And the syllabus doesn't actually name these 3 parts, so I'm not sure how absolutely vital they are. So I'm just going to include 3 parts that are found specifically in prokaryotes that are often not necessarily found in eukaryotes. Flagella we'll talk about as like a tiny exception. But some prokaryotes have these additional structures, which are a capsule, flagella, and pili. So the capsule is basically allows for kind of anchoring to other cells. The flagella is for movement, and pili is for DNA exchange. So these are just kind of features. They're not necessarily on every prokaryote, but you do see them quite a bit in prokaryotes. So these are found in many, but not all prokaryotes. And if I were to kind of draw a bacteria and show what they look like, the capsule is kind of a thicker outer layer that usually lets it stick onto surfaces better. The flagella is a nice tail for it, and pili are these little like larger than cilia projective hairs, and they also can join together and form like a little tube so DNA can be exchanged. Again, not mentioned by word on the syllabus, so that's just kind of a little bit of a covering ourselves in case that comes up. What I'm going to do next is go into a handful, or 6 organelles that are found in all eukaryotes, but not in prokaryotes. So kind of done with prokaryotes. So we'll start with the most important organelle, which is the nucleus. And the nucleus has its own envelope, which is its own membrane, and it has its own nucleolus as well. So the general nucleus contains DNA. The purpose of the envelope is to have pores to let mRNA out. And the nucleolus makes ribosomes. I don't have a particularly great way of clarifying that it's the nucleolus that makes the ribosomes. I'm just going to underline that. But that's just an extra little function of the nucleus itself. This is found in all eukaryotes, but not in prokaryotes. If you were to see it on an image, you can, depending on the quality of the microscope, potentially see the pores. If you're ever drawing it, I would recommend drawing the pores. A darkened area for the nucleolus, and then there's usually just kind of condensed areas where you'll see that represent the DNA. So that would be how you would see it represented on a microscope image, and also kind of a good guide for how you could draw it as well. All right. Next, I'm going to pick up on all of the organelles that are linked to ribosomes, so all of my kind of protein processing organelles. So in eukaryotes, that can be found on the endoplasmic reticulum. So next up, we're going to have the rough ER and the smooth ER. So rough ER is particularly going to be involved in the transport of proteins. Smooth ER is going to be involved in the making and transport of lipids. Like so. Where are these found? Again, all eukaryotes. Generally the rough ER, so again I'm going to refer to my image. So you can usually see it looks like little lines. If you were drawing a diagram of it, it would be worth drawing some ribosomes on it. Sometimes you'll see them kind of connect looking like that. For the smooth ER, it says a lot of times you can't necessarily, they're like stacks instead. So you can't necessarily see, they tend to sit more vertical. So a lot of times they'll look more like a set of kind of circles. I've never really seen smooth ER drawn like that. Usually you'll just see it represented as like, looks the same as rough ER without ribosomes. But on electron micrograph, I think it looks a little bit different. So covering our cells will show that. So again, folds with some ribosomes on it if you're drawing it as rough ER. Okay. So that's for transport within the cell. If we want to actually move it out of the cell, it is then going to go to the Golgi apparatus. So the Golgi apparatus is going to modify and package and ship proteins and lipids. And the packaging, when we say package, that's going to be into vesicles. So they're going to be packaged into vesicles. Again, that is going to be found in all eukaryotes. And this is going to look distinct from the ER because it looks like stacks that are unconnected. And they kind of have this varying in size where you'll usually see a smaller and then kind of larger. And then you'll also see the kind of vesicles coming off of it. So they do actually look unique from ER, so you should be able to differentiate them based on whether they're kind of all one length and kind of connected, or whether you can clearly see a stacking and a size difference. All right. Another super important one that's going to be in all living organisms is the mitochondria. And this is really important to remember that it's in all living things, because it's, in all eukaryotes, because it's an easy trick to think chloroplasts in plants, mitochondria in animals, but plants have mitochondria as well. So this is another organelle that's in all eukaryotic organisms. Its purpose is going to be aerobic cellular respiration, which we'll do more on later. So there is an anaerobic form, which is called glycolysis. So that's basically involved in the making of ATP. Just gonna move that next to it so that I'm running low on room on the other end. So making of ATP, again, all eukaryotes. And if you see it, so a lot of times we might draw it, and we'll draw it looking kind of like that. If you actually see it on an electron micrograph, it tends to just have lines through it. So just a kind of an example of that is when you see it, see if I can get this image okay. That's the mitochondria. So it looks kind of circular with the lines through it like that. So that's just kind of a bear in mind that it's not always going to look like the little squirrely inside. So if you see anything that looks like that, recognize that that is looking at a mitochondria. All right, two more. I'm having to do these fairly brief. I'm running out of room. So the next one is a cytoskeleton. And that's kind of, there are components of it in all eukaryotes. It does look a little bit different in plants versus animals. So animals tend to have a more complex cytoskeleton because they don't have a cell wall or the vacuole. But you will see some microfilaments and so forth in plants as well. So cytoskeleton is going to basically maintain cell shape. And it's microfilaments and tubules. And it's going to be in all eukaryotes, but it is going to be more complex in animal cells. So it is going to be most complex in animals. And that, again, that just relates to those cells not having a cell wall. So they tend to have that as kind of an extra kind of support. And a lot of times it just looks like thin kind of tubes and so forth throughout the cell. Sometimes they're so thin they're not really even visible. But if you were to see them on a micrograph, that's what they would look like. All right, and then lucky last, and this one's kind of a weird one to put specifically in our they do all have it, but there is quite a bit of variation, and that's going to be vacuoles. And so vacuoles are going to be for the storage of any materials, so they can store food, waste, water. And again, these are going to be in all, but we do need to make a distinction that they are specifically large in plants. So in plants, they're going to be a large central vacuole. And then in animals, they're going to be small and diverse. And so instead of having one that kind of stores everything, you're going to see there's a lot more of them, and they can have slightly different functions. In terms of what do they look like, sometimes basically if they don't have much food in them, they may look hollow. If they have food in them, they may have a little bit of shading in them. So it kind of depends on how full it is and what's inside of it. Okay, so that is our last everything one. I'm very tempted to cheat and erase just so I'm not writing so tiny. You probably have more space than that. So the last three, so I'm going to erase, but you keep going. But I'm going to be like writing super small, and it's probably harder for you to read. So the last three things I'm going to put on here, I'm just going to at least erase these last two. That'll be a good compromise. They're kind of dumb anyway. Okay, doke. So the last three things are specific to either plants or animals. So with plants, we want to add plastids. So in particular, the chloroplast and the amyloplast. So sometimes those will be included together. It's just kind of a membrane-bound kind of pigmented area. And so what is their function? Of course, the chloroplast is going to be the site of photosynthesis. And then the amyloplast is going to store starch inside of plant cells. So these are both things that are specifically found only in plant cells. And you can generally see a chloroplast. They'll usually have something like this, and you can actually see the kind of flattened stacks inside of them. So have a look on kind of a micrograph to see those kind of stacks is how you could make those kind of out in that. One thing that's found specifically in animals then is going to be lysosomes. And lysosomes are basically just a container of enzymes. So it's actually a type of vesicle. So it's a vesicle with digestive enzymes, like something 40 different enzymes inside of it. They can break things down. So it can break down food, organelles, kind of any number of things. So it just has that ability to hydrolyze. So it's hydrolytic enzymes, so specifically enzymes involved in breaking things down. These are specifically found in animal cells. There might be some in fungi, because again, they don't photosynthesize. So some fungi might have forms of lysosomes, but they're very prominent in animals. And they usually would look like a vesicle, but you could kind of see scattered, like a less transparent one, because you could see that there's stuff inside of it. Okay, and then lucky last. Last one is a couple of different things. Again, I'm going to kind of throw together of just some things that are generally unique to certain animal cells. Well, first of all animal cells have centrioles. And then the other that some have is cilia, and some even also have a flagella. So those are some kind of other unique adaptations primarily found in animal cells. There are some plant reproductive cells that will have some cilia, but it's pretty rare. Centrioles are going to be for cell division. Plants and fungi do have the ability to produce spindle fibers, but they don't have unique organelles for it. So the unique centrioles that do that is specifically a feature of animal cells. And then cilia is going to trap food, which is again why we generally see it in animals. And then of course flagella is for movement. And again, animals tend to have more kind of movement than plants. These are going to be usually animal cells. There are some examples of them in some fungal spores and the like. The centrioles do actually kind of look like two tubes that you can kind of see near each other. And of course, we already talked a little bit about what flagella look like. So the centrioles would be visible as kind of a pair of tubes. If you were looking at it kind of through a cross section, they might look like this kind of next to each other, usually near the nucleus. Okay, so that was my exhaustive list of organelles that I cheated and did erase some. Again, for right now, you're good on the fairly one-line definition for what the function is. Some of them will go into more detail, not all of them. So when we get to B2, we'll go into a little bit more, particularly on the Golgi, the mitochondria, the chloroplast, and the nucleus. So there will be a few that we'll kind of discuss more. And then some of these you do just need to know kind of straightforward what their overall function is. And we're not going to necessarily go into tons more detail about it. Okay, over here on our remaining little area, two things that we're going to add, and I'm going to erase my chart just so that I don't have to write it way over on the side and you have to squint as much, but hopefully you have your area over there. So the two remaining bits, one more that's SL, and then there is one tiny HL thing. So the SL thing is to look at the idea that we're kind of presenting this idea that this is what all cells have. So all plant cells have this, all animal cells have that. But in multicellular organisms, that's not necessary for every cell to have every component because they may be carrying out a specialized job. So to show that the same organelles are not necessarily found, we're going to look at 4 examples of atypical eukaryotic cells. And we're going to, like I said, look at 4 examples. And the examples are all related to not following the one nucleus rule. So we're going to look at 2 examples that have more nuclei and 2 that have less. So we're going to look at 2 that have more than 1 nucleus per cell. And basically, I just want you to know these examples. We did draw it in class, but I'm not going to redraw them. They're not that exciting. So first one is going to be fungal hyphae cells, which are kind of like the roots of a fungus. And they are long and skinny with lots of nuclei, like they basically lost their dividers. So instead of being individual cells, they're now one long cell, and then they still have multiple nuclei from when they were once multiple cells. Very, very similarly is skeletal muscle in humans and other mammals. Similar concept, it is long cells that in order to meet the needs of the long cell will have multiple nuclei that are actually usually pushed to the side. It gives greater elasticity and flexibility than having lots of short cells. Okay, so that's more than one nucleus. The next exception is to have less than one nucleus, so lacking a nucleus. And there's two examples we looked at that do not have a nucleus. So one is in plants, which is phloem sieve cells. And then essentially in order to kind of better facilitate the movement of sap, they actually have removed their organelles to create a better kind of transport area. But to make up for that, they actually rely on companion cells. So they still have a neighboring cell that allows it to do its job for it. And then the other example is red blood cells in humans and other mammals and other organisms that have red blood cells, and they basically they're almost not a cell, so they can't reproduce on their own, so they don't meet the criteria of a cell. So they're basically like a product of a bone marrow cell. So they're like an atypical cell for sure, but that basically increases the surface area. And the greater the surface area, the closer the hemoglobin is. So that helps it transport oxygen more effectively. And they just die after 3 or 4 months and get recycled for parts. So they definitely have some atypical features about them. And then why is this possible? And this is possible because they are multicellular organisms. So they can have some cells that don't do everything because they can have other cells that do help, like the companion cells, or like in the example of the red blood cells, you have bone marrow cells that can help in making them. So possible due to specialized cells. And that happens in multicellular organisms. So because they have many cells, they can specialize. And then this is just a tiny dot point here. It's going to get picked up on way more in other topics. But when we talk about specialization, the specialization of cells is due to gene expression. So the expressing of some genes and not others. So they use the genes that are appropriate for their function. Okay, that's actually the end of all SL. So if you're SL, goodbye. If you're HL, we need to do one more thing here, and then we still gotta do viruses. So last little thing for HL is the endosymbiotic theory, which you can put under yours in your little side column. I'm gonna move over here, and we're gonna do a pretty short version of it. Probably the most important thing is that you actually know the evidence for it. So endosymbiotic theory. So I'm going to show kind of the end point. So here's my massive early eukaryote. And we say, well, how did it become a eukaryote? So interesting ideas that essentially some like folding of the membrane around the nucleus to create kind of the DNA container, and then some folding of the plasma membrane to create endoplasmic reticulum and things like that. So a lot of that was thought to actually be just kind of enveloping of its own membrane. But then it was hanging along and it engulfed some prokaryotes. So here is one prokaryote. Actually, and I'm just for accuracy's sakes, sorry. I'm going to draw that in black. We'll talk about why in a minute. So it engulfed this other prokaryote. And then eventually it's going to engulf another one. I'll go ahead and draw both of them. And then instead of digesting them, they retained function inside of them. So they engulfed it, and then they retained function. And those functions were helpful, which actually made that cell the most fit and happiest cell of all time. So he's happy because he got these little helpful organelles living inside of him. And so he's going to outcompete other cells. And so then over time, they have now become a part of all eukaryotes, and they have now become the organelles, mitochondria, and chloroplasts. So that's endosymbiotic theory. The key thing is that it's the early eukaryote that did the engulfing, and then who he engulfed is the prokaryote. And instead of digesting them, those prokaryotes remained inside and performed helpful cellular functions, which created a benefit or an advantage. Therefore, natural selection, they stay inside. So he's happy and will add increased fitness as a result of having those organelles. That's why he's happy. Okay, so then going back to kind of where we were at in the A1 section. Oh, and I don't want to forget to make a little HO. Okay, and so then the question is, what is the evidence of this? So the evidence relates to things that mitochondria and chloroplasts have that are unique. So, and I tried to draw it, forgot to draw it on him, so I'll add it there. There we go. And chloroplasts. So first I drew that they have their own DNA, and in particular they have circular, so they have their own DNA, and that DNA is circular, which is, we remember, is a common thing in prokaryotes. They also have their own ribosomes, and those ribosomes are 70S sized, so they are prokaryote sized. While we're on that, we'll just make note that they are the approximate size of a prokaryote. So the organelles are about the same size as modern bacteria. The other thing they have is a double membrane. And that outermost membrane is thought to actually be the eukaryote membrane. So the outer membrane actually has more features in common with eukaryote cell membranes. So they have kind of two membranes that look different. And the other thing is that they have the ability to self-replicate separate to the cell. So they can replicate on their own, not just when the cell replicates. So definitely need to know the endosymbiotic theory, know your evidence, be really clear on who engulfed who, so eukaryote engulfed prokaryote. It is worth noting that that happened at separate times, so it's generally accepted that the mitochondria happened first, and then another event happened that led to chloroplasts in some organisms. All right, that's everything cells. Viruses next. Okay, last part of this topic is viruses, and this is another part that is HL only. So this is only higher level. So just to try to organize it a bit different, I'm going to do 4 viruses. 3 of them you definitely have to know the structure of. On influenza, you have to know a bit about how it evolves. You don't actually have to know the structure, but because of my like to draw consistently, I will draw the structure, but I'll make a note you don't have to know it. But essentially, you need to know the structure of bacteriophages, HIV, and coronavirus, and then you have to know a little bit of special information about each one. So I'm just going to work it that way. I kind of drew my lytic and lysogenic really tiny, so if you want to use a little bit more space on the bacteriophage, I kind of did it even, but that one probably has the most information about it, so totally fine to go for that. So we are going to look at 4 significant viruses, and we're going to basically learn all our viral material through these 4 viruses. Okay, so up first we have bacteriophage lambda. So I'm going to try to draw, you're going to do 4 in total, so I'll erase my board once. So I'm going to try to kind of, just to give ourselves a sense of space, this is like half. Okay, so bacteriophage lambda is in some ways simple because it doesn't have an envelope, but it does have a bit of an interesting shape to it. So its caspid looks kind of like this. Then it has this little tail sheath like this. Okay, so all of that is kind of protein capsid. Then coming off of here, we're going to have our little spikes. So those are usually help to actually allow it to latch on to its target cell. And then it needs to have some sort of nucleic acid. So rather than drawing like what's common to all viruses, we're just going to draw it in all 4 and you'll see that. And so this one actually has double-stranded DNA. It is possible to have single-stranded DNA, double-stranded RNA, so there are some variations, but that is an example that is actually double-stranded DNA. And so then let's name this guy. So this is, sorry, I should have given enough room for that. This is bacteriophage lambda. Okay, so that's our first virus we need to know. In addition, we need to know who it infects and what its interesting facts are. So it infects bacteria, and in particular, it infects E. coli bacteria. And then for each of them, what's something that's interesting? So we'll say interesting because ... And so what's interesting about this one is it can undergo both a lytic and a lysogenic life cycle, whereas a lot of viruses are solely lytic life cycles, or they have some ability to undergo a lysogenic life cycle, but then they generally don't go straight into lytic. They always do that. So that's what's kind of unique. So it can do a lytic life cycle or a lysogenic life cycle. Okay, so lytic or lysogenic. So I'm going to draw it in class. I drew it as like 7 steps. I'm going to condense a couple, and I'm not going to worry about the official names. There's different terms for the steps in different books, so there's not like you need to memorize the names of the steps. You would more need to be able to describe the steps. So I'm going to, like I said, kind of consolidate it a little bit for the sake of revising. Okay, so step 1, the little bacteria, he's going to have to come along and he's going to have to attach on. So that's going to be his first step, is to be able to attach. There we go. So step 1, attach. Okay, then he's going to insert just the DNA. So the rest of the virus will stay outside, but the DNA is going to get inserted. So there's the insertion of the DNA, and the virus is just going to stay outside. Okay, so then I'm going to do, like I said, a little bit of a simplification. So this is insert DNA. Then it's going to copy the DNA and transcribe it into messenger RNA. And I'm just going to kind of show both of those as one single step. Try to keep that looking vaguely like a circle. Instead of, you could draw it as 2 separate steps, but we're just going to keep it simple. So it's going to copy DNA, and then it's probably also going to copy some messenger RNA coming off of them as well. So replication and transcription. So replicate and transcribe. Once it's transcribed and has mRNA, it's then going to be able to build new viral parts. So now it's going to build little capsid heads. And little tail sheaths and little sticky little prong guys like so. So then it's going to translate viral parts. And then it is going to assemble, and I'm actually going to draw the last, again, you could draw this as two separate steps, but as the viruses start to assemble inside of it, they are going to essentially crowd it out. So because of the kind of volume of viruses, they are going to start to take up so much space that that causes it to burst, freeing the virus. And then it will go and infect another cell. So that's going to be assemble and burst. So again, you could break it up into more or less steps. I said lice. But the basic idea you need is that it's just the nucleic acid that gets inserted that has to copy those instructions, use the instructions to build virus parts. Once they assemble, it puts strain on the cell membrane, causing it to burst. And it is this bursting that's generally going to be associated with symptoms. So once the cells start lysing, it's going to cause damage to cells, which is what creates symptoms. It can also go through a lytic cycle, and if it goes through a lytic cycle, it kind of goes through the same first couple steps, but here it's going to change. And so now that viral DNA is going to integrate in with the host DNA. So it's going to integrate. And there is a term for that that's actually called, when it's integrated, it's called a prophage. Don't really think you need that term, but it's always good to have extra words just in case you're writing about it. And then if that cell goes through cell division, it will get copied along with the host DNA. So now, if that cell goes through division, now there's two cells that have it. And again, I haven't done a very good job of drawing E. coli because I'm just drawing a box, but just a reminder that each one of these is like a little E. coli cell. So just a reminder that is a cell and not a box. So it will spread through cell division, and it can continue to replicate with divisions. So it can continue to do that, or it can detach and enter the lytic cycle. So sometimes viruses can stay in kind of a dormant lysogenic state for a long period of time, and maybe exposure to stresses or something like that can cause it to pop out and detach and then enters into that next life cycle. So that's our interesting fact about bacteriophage, is that they can do the lytic or lysogenic cycle. Okay, next virus, the coronavirus. So it also has a capsid. It has kind of a strange capsid that's like really kind of tightly wound next to the RNA. So here's kind of the RNA, and it's kind of like linked onto the capsid. So a slightly strange kind of capsid structure. And then this also has a viral envelope, which is actually made up of like a phospholipid bilayer. So it ends up looking kind of similar to the cell membrane of its host. So essentially when it formed, it actually took that viral envelope with it, or that host envelope, and now has its own kind of cell membrane. And then it has little small protein spikes. So those are its protein spikes. So protein spikes, capsid, and then we said what's unique about this one, and actually all of the other ones we're going to draw, is that it actually doesn't have DNA but has RNA instead. Okay, so who does it infect and why do we care? So we should know some of this. It infects respiratory cells, and we think it possibly a range of mammalian respiratory cells based on the fact that it's believed to be due to coming from bats. So at least it looks like it can infect at least some other hosts, not just humans. And it's interesting for two reasons. First of all, is an example of zoonosis, which means it crossed species. So we got it from most likely a bat species. And also interesting because, shocking revolution, it caused a pandemic. So that's actually fairly rare for viruses to cause a global pandemic. So that's a big deal. So while we're on this one, we're going to look at something that's not necessarily specific to coronavirus, but just a general thing you have to know about viruses, but I had Spot here. So then our next question is, where did viruses come from? So in terms of like zoonosis, of course, is one cause, but where did viruses like initially come from? So like how did they evolve? Why do we have them in the first place? So first of all, the fact that we have like DNA versus RNA, so they have so many differences. So if you look at these two, they're so different. So the fact that they have many differences means that we don't think they actually evolved together. So they most likely evolved separately. So they might share some common features, like protein spikes are helpful, but that's not because they actually evolved kind of from a common ancestor. So evolved separately, no common ancestor. And so just a nice little revision of our bio terms. When something evolves separately with no common ancestor, we call that convergent evolution. Okay, while we're on the topic, so okay, they evolved separately, but we're still like, where did they come from? So there's actually three kind of origin hypotheses. And we're going to go into it in like the world's simplest detail, because I would be surprised if you got asked much about this, but it's just to recognize that there's not like one explanation, that we don't actually know. And some of the books cover all three of these, some of them only cover two of them, so I don't think there's like a set thing that you have to know about, but just be able to recognize that there's not an established cause of them. So one hypothesis is that viruses predated cells. So that actually before cells evolved, there were viruses and cells actually started as viruses. That doesn't really agree with the rest of our hypotheses in terms of primordial soup. So that's not likely to be the case because it doesn't line up with all the other stuff, but that is a hypothesis. The next one is called a regressive hypothesis. And that actually says that what happened is they were once cells and then they lose function and become viruses. So they're basically like used to be cells that have simplified. They have lost things over time. So it was once a cell, but then it's lost some features. And then the opposite of that is actually called a progressive hypothesis. It's also sometimes called like an escape hypothesis, which basically says it's like a part of a cell. So it would just be like DNA or something like that escaped from a cell. And then it actually gained structures and became a virus. So a lone strand of DNA or RNA escaped, and then it developed its own capsid and spikes, and now it's a virus, versus like whole cell kind of started to lose some things, and now it's a virus. It's not anything I would like spend a lot of time losing sleep over this idea. The long and short of it is we don't know. So different ideas as to kind of where exactly they came from. The one thing that seems to have the most kind of traction is that however it happened, it was not a single ancestry event. So it is possible that it's different. Some of them actually maybe were one cell. Some of them might have actually come from a bit of DNA. So it could possibly be even like a combination of both of these because of the fact that it didn't happen the same way. Okay, so that is viruses. Sorry, half our viruses. We got 2 to go. But the last 2 are simpler because well, yeah, we don't have to do this whole lytic lysogenic thing. Okay, so HIV, and HIV is interesting. We will come back to it a bit later on in the course. So when we get to the immune system, we'll talk a little bit more about it. So we're doing kind of intro to it, but it is something that will reappear in the course. Okay, so, and you'll notice these last few look quite similar because they're all human virus examples. So, and they actually, yeah, so they're structurally more similar as well. So this one has a bit more of a normal kind of capsid that's not quite as tight with the nucleic acid. This also has RNA, but it actually has 2 identical strands of RNA, which is a little bit weird. So they have the exact same genes and everything. And then this has an interesting other feature is that it has an enzyme kind of in with it, and that's reverse transcriptase enzyme. Okay, and then similar, I don't know why I drew that as orange when I also drew this, but that's fine. It also has an envelope. So it also has a viral envelope. And then it has slightly more complex spikes. So they're actually glycoproteins. So they have kind of those longer carbohydrate chains to them. So slightly longer spikes, but similar in function. So glycoprotein spikes. Okay, so that little bit of something extra over the coronavirus was that it does have that enzyme in it as well. Let's look at what does it infect and why is it interesting. So it infects human helper T cells, which are white blood cells. So when we get to the immune system, we'll talk more about what helper T cells are and their specific role, but it's part of our immune system, and it's actually a really important white blood cell that kind of activates a couple different loops. And then why is it interesting? 2 reasons. So one reason why it's interesting is because it is a retrovirus. And that is where that reverse transcriptase comes in. So what that actually means is it has RNA as its nucleic acid, but then it actually turns that RNA into DNA inside the host. And so then the RNA becomes DNA, and then it becomes RNA again for normal transcription. So RNA becomes DNA and then gets transcribed inside the host back into RNA. So it's a bit of a strange pattern, and this is reverse transcription, and that's where it includes its own enzyme for that. So it inserts its RNA and the enzyme. It turns into DNA inside the host. That DNA gets copied and then transcribed. So a little bit of an extra complication. The other thing that's interesting about it is that it evolves rapidly. And it evolves rapidly due to a process called antigen drift. Antigen shift. So you can tell I just made this today and I still got those terms wrong. So be really careful because they are similar sounding. So antigen shift. So shift for, I'm going to just double check my source material and make sure that I am happy with that assumption. So yes, so shift rather than drift. We will go into both of those in just a minute. So this one is, hold on, no, drift. See? Drift sounds a bit more slow, so you're going to drift along. So this is the slower process. So sorry, very confusing because they sound very similar, but this should actually be drift because it's going to happen a bit more slowly. So what happens is we have our virus. Here we go. And it's going to have its RNA, and then it's going to have kind of very specific antigens, which is what these glycoproteins are going to act as because they're kind of identifiers, and our immune system identifies it by that. But then inside the host, it's going to undergo a series of mutations. So it's going to go inside the host. And then once it's inside the host, it's going to have some mutations occur. So slowly accumulates mutations. And then some of those mutations, what they're going to end up doing is they're going to end up changing the spikes. So now I have a whole new genome, and now I'm going to have different spikes that have resulted from kind of an accumulation of mutations. And the problem is those spikes are no longer recognized by memory cells. So that can cause problems for the individual, because what's happening is over the time of having the virus, they're not building any immunity or ability to recognize and fight it because it's evolving and changing. And so their own immune system kind of has to start over all the time. So your own immune system is fighting like different versions of the virus constantly. Okay, so that is, sorry, I keep forgetting to label these. I don't even think I labeled the last one as coronavirus. I'm so sorry. If you could go back, I erased it already. I should have labeled them at the top, but make sure to go back and label that as coronavirus. Hopefully the caused a pandemic will remind you, but go ahead and do that because I forgot, sorry. Okay, and then last one is influenza, and you do not have to know the structure of influenza. You do for the other three, but you have to know how it evolved. So I thought, fun, let's put it in our chart. But you don't have to know this. So it is also has a capsid. It actually has like RNA fragments. So it works a little bit different. Instead of having like a full RNA connected sequence, it has fragments of RNA, and also has its own enzyme that then helps kind of put those together, like a polymerase. We're not going to draw that because you don't have to know this. I'm already drawing unnecessary stuff, so I'm not going to also draw unnecessary and confusing stuff. And then it's also going to have some spikes. It actually often has kind of a couple of different spikes that relate to like different strains, and that actually relates to what we're going to talk about. So a lot of times it will have a little bit of variations in the spikes that it has. So this is influenza. And then I'm just going to write do not need structure. Okay, who does it affect and why is it interesting? So this is also going to infect human respiratory cells. So has the potential to cause secondary pneumonias and some characteristics that are similar to COVID that way. Why is it interesting? So there's only one thing you really need to know about it, but it is listed as interesting because it also evolves rapidly, but it evolves rapidly by a different mechanism. So also evolves rapidly. And this is why I kept getting them confused, because they sound so similar. Instead of drift, which you're drifting along, it's like shift. So it's a faster change. So this is antigenic shift. And they are quite different in terms of what happens. So this time, one host actually gets invaded by two different flu viruses. So it would be like, here's version 1, here's version 2. So over here, this one comes in. I'm just doing it in the same color to make it slightly easier to recognize what's going on. And this one comes in. And it actually both come into the same host at the same time. So the host actually gets invaded by two viruses. So one host is invaded by two strands. And then in there they quickly go through DNA exchange. So quickly exchange genetic information. So what ends up happening is they kind of will share genes. So they'll say, here, have some of mine, have some of mine, and the DNA will kind of splice and reassemble. And then by the time it's actually making viruses, what's ended up happening is we end up with like a whole new virus relatively quickly. So it quickly forms a unique virus. So by exchanging that DNA, it's very quickly providing that new variant. And that new variant is once again not recognized by memory cells, including those produced by vaccination. So then when this spreads through the public, those that have been vaccinated are kind of vaccinated for maybe one of these forms, and suddenly we've got that form. So it's not compatible with where the previous vaccines had covered it. And so that is why we have to get flu vaccines all the time. And a lot of times they actually are like trying to predict what changes might happen based on known flu viruses and kind of predict how this could occur. So it's a little bit of an imprecise process, but essentially trying to figure out what could be coming, and then sometimes they get a pretty good idea, sometimes it's not as effective, and then by the next year, based on what did happen, you have to get a new vaccine that covers you from that one and has some sort of kind of predictive powers. So antigenic drift versus antigenic shift. And then it could continue to also undergo drift, but that shift is that two joined together to one, and then the drift is the more slow change just by accumulating mutations. So, all right, that's everything. So thanks so much. Bye.